All right, everyone, if you're joining us a little early, we're going to give it just a little bit until everyone gets in, and then we'll get things kicked off. Thank you.
I love watching the names pop in on the screen. There's a nice, lovely mix as always of people who have been here for every episode and some new names as well. So welcome to everyone.
All right, I think that's a good starting point for us. Thanks everybody for being here, and welcome to our 25th episode of Ewing's U. And thanks for your flexibility on this one. This is an episode we had slated for January, but suffice it to say we had to pivot a little bit. But here we are rescheduled, and so grateful for this opportunity. For those of you that don't know me, I'm Piero Spada, and I'll be your host today. I'm also co-founder at Little Warrior Foundation, and more importantly, father to 14-year-old little warrior Lucy. If you're part of our core contingency, aka the Little Warrior Fight Club, thanks for being here with us since day one and making this a place for positive change. And if you're new, well, welcome to Ewing's U. It's our webinar series where we hear from leading sarcoma experts and discuss the cutting edge of care and science behind Ewing sarcoma. Today, we'll be hearing from one prominent expert from the NCI and Dr. Christine Heske to discuss the recently opened PEN-866 trial.
You ready for me?
Yeah, nope. There's just like a little bit of a lag on the slide. Sorry. Briefly, Dr. Heske is a pediatric oncologist and physician scientist with a translational and clinical research program focused on improving treatments for sarcoma. Her goal is to improve outcomes for patients with pediatric sarcomas by understanding mechanisms of resistance and identifying and evaluating new therapeutic targets. Major projects in her lab have focused on characterizing and targeting biological vulnerabilities related to tumor metabolism, DNA repair in pediatric sarcomas, such as Ewing sarcoma and rhabdomyosarcoma. In addition to conducting preclinical laboratory research, Dr. Heske is invested in translating promising laboratory findings into early phase clinical trials for young patients with sarcoma. But before we kick things off with Dr. Heske, just a quick reminder that Ewing's U is a webinar production of the Little Warrior Foundation. We are a National Childhood Cancer Research Foundation with a laser focus on finding a complete and permanent cure for Ewing sarcoma. We were founded in April of 2020 and have now granted out over $2 million in high potential therapies for increasing survivorship and decreasing toxicity. And in order for us at Little Warrior Foundation to make programs like this a reality, we rely on the support of the little and big warrior community alike. We appreciate all of your support so that we can continue our mission of bulldozing our way towards more effective and less toxic therapies and continuing this vital work for kids battling Ewing sarcoma. With that, I'm going to hand things off to my co-host, Emily, to introduce herself and touch on a couple of housekeeping items.
Hello, everyone. I'm Emily McFadden, one of the co-founders of Little Warrior Foundation and godmother to Little Warrior Lucy. So excited to have everyone here today. A few housekeeping notes before we hand it off to Dr. Heske. Please use the Q&A function of Zoom down in the lower sidebar to ask questions as they strike you. We will save all the questions till the end and answer them there. As a reminder, we can't discuss any specific cases or provide direct medical advice in this format. So if you can keep your questions general, that would be super helpful. We're really excited to have everyone here today. And with that, I will kick it off to Dr. Heske.
Thank you so much. Everyone, thanks for joining. And I'm just going to try again to share my screen because we tried this a few times. I think it's going to work. Yes?
Nailed it.
Yep.
All right, good. So thanks everyone for joining again. It's really a pleasure to be here to talk about this clinical trial today. And as Piero referred to, it's been a little bit of a long journey to get to this webinar, originally scheduled for January, but we're here and I'm really excited to kind of talk about the work that we've been doing. Okay, and what I'm specifically going to talk about today, let me just get my laser pointer going. Okay, is this phase 1/2 study of 3 drugs, 2 maybe known to some of you, vincristine and temozolomide. Those are kind of standard therapies that we use. in combination with a experimental agent called PEN-866 for adolescents and young adults with relapsed or refractory solid tumors. And so we abbreviate this very long name by calling the study VTPEN, and the NCT number is right there in case anyone wants to look it up on clinicaltrials.gov. And so I just kind of wanted to back up, but before I talk specifically about the trial, to talk about kind of the history of pediatric cancer progress and therapies in general. So as Piero mentioned, I came to this field, I'm purely MD trained. I had no bench experience when I entered medical school or residency or fellowship. And during my training, it just became really clear to me that there was such an unmet need, particularly in the area of pediatric sarcomas. And so I come to this kind of with a clinical bent when I think about kind of what has happened in the history of therapeutics for our patients. And I think of it in two pieces. The first is really advances that we've made in the understanding of the biology of pediatric cancers. And the other is the advances in the treatments and clinical outcomes for our pediatric cancers. And what I have noticed, and this is certainly not a new idea, many other people have noticed this as well, but is that in the last decade and a half, we've made massive advances in the understanding of pediatric cancer biology. We understand so much more about what drives these cancers, the mutations that they have, the variety of different sort of subtypes of each of these. But unfortunately, and in particular for pediatric sarcomas, it has not translated very well into advances in treatments and certainly not into clinical outcomes for many, many of our patients. And so there is a huge disconnect. And this disconnect is really what drives kind of me to do the work that I do. And so, you know, this begs the question, why is there such a discrepancy? And I think that one of the reasons is that we have so many, if you think about it, kind of all of the different therapeutics or potential targets for different cancers on this side, very, very few of these actually make it to the clinic, and very, very few, even fewer, make it to the clinic for children and young adults and adolescents. And so I think, full disclosure, that the today here was in January. I didn't update this slide, but I think it's not changed in a couple of months, is that there's just 25 more or less phase 3 clinical trials testing a new treatment for any pediatric cancer patient in the United States right now. So if you think about all the clinical trials that there are for adults, this is actually a really tiny number, just 25 for all pediatric cancers. And I think this is due in large part to a number of systemic challenges that we know exist in pediatric drug development. You know, and frankly, I think that the current system that we have disenfranchises children because it doesn't capitalize on the timing that adult drugs, you know, can use to go through the development pathway. Usually, we have to wait for a drug to be tested in adults before we're allowed to test it in children. There's, you know, in the mind of a lot of pharmaceutical companies, there's more risk in testing drugs for children. And then the financial incentives of developing a drug for an indication that doesn't affect a lot of people also kind of plays into this as well. So what I have experienced kind of in my own practice, and I think many other clinical trialists as well, is that potentially active financial orphan drugs often have no path forward. So if they're stalled at the level of adult drug development, even if they look really promising in early models of pediatric cancer, we have no way to access those drugs. And so what this ends up causing is a really huge lack of new drugs or new agents that have shown sufficient preclinical activity in pediatric cancers that are then clinically available to study or test in children. So while we may have a lot of interesting insights from the lab, very few of those actually make it into trials. And what this means is that when we lose the potential to test new agents, we lose the potential to improve outcomes for patients, because no matter how much we know on the basic science side, if we're not getting that into the clinic, it's not helping our patients. So kind of with this as the background, I know that's a very depressing background, but with this as the background to this story, I just kind of wanted to touch on the study timeline for this trial that I'm going to tell you about. So we did this preclinical work about a decade ago. I was actually still a fellow in the lab when we did this, and we published the work in 2016. And then we worked with a drug company who ran this first in human clinical study in the late 2017 into 2020. And during that time, we developed a combination study, the one that I'm telling you about for adolescents and young adults. And then, unfortunately, about a year after we finished the development, the industry sponsor filed for bankruptcy and the drug became unavailable. And so I'm going to kind of tell you each of the little pieces of this story in more detail and then where we ended up today. So I'll start with the preclinical laboratory studies, and I'll try to kind of go quickly through this because it's not necessarily the most important part. So the first thing I kind of want to mention to you is the class of drugs that this experimental agent is called a topoisomerase 1 inhibitor, or a TOP1 inhibitor. And what TOP1 does, it's an enzyme that like all normal cells have, and it helps when cells are replicating their DNA to kind of relax the coiling of the DNA. So if you imagine that if you had like two pieces of a rope and you pulled them apart, you'd get it would become harder and harder to pull them apart because you're getting sort of twisting on the back end. This enzyme makes a little break in one of those pieces of rope and relaxes it so that you don't get overcoiling. And this happens in normal cells, it happens in cancer cells, but this class of drug agents actually acts on that relaxation step. So it prevents the two strands of DNA from joining back together. And what you end up with is little breaks all over the DNA, and that can cause a cell to die. And that's the whole kind of philosophy behind this class of agents, the TOP1 inhibitors. And we know TOP1 inhibitors in the pediatric sarcoma world because there's one that we use very frequently, and that one is called irinotecan. It is highly active. It's commonly used as part of multi-agent chemotherapy regimens. It's used in the upfront setting for certain sarcomas. It's used in the relapse setting for others. And we know that when you expose cells for a prolonged period of time to TOP1 inhibitors, you get really, really positive responses. You see tumor shrinkage or tumors completely regressing and going away, both in models and often in patients. But we also know that irinotecan has a lot of limitations. It is what we call poorly bioavailable. So what that means is that even when you put it into the patient, the amount that actually gets to the tumor in an active form is a lot less than you gave the patient in the first place. And this is because it enters the body in an inactive form, and it has to be converted into that active form before it can do anything in the tumor. And that conversion itself is not a very efficient conversion. We also know that it has a number of toxic side effects because, like all chemotherapies, it works kind of equally in the normal cells as it does in the cancer cells. And so it can cause the same sorts of side effects that many chemotherapies do, like low blood counts and hair loss and nausea and vomiting and that sort of thing. And particularly, we see these come up in drug combinations because chemo has all these overlapping side effects. And so because it's pretty well known that irinotecan has a lot of limitations, there have been a number of recent attempts to make it better, to make it less toxic or more active or convert more quickly, et cetera. So some of you may have heard of a drug called Onivyde. This is a nanoliposomal form of irinotecan. There are a couple of trials actually running with this drug right now. And this actually just delivers kind of the irinotecan in a different formulation in these nanoliposomes, which are like very, very small particles. But at the end of the day, it's still delivering irinotecan. And so all of the limitations that come with normal irinotecan are also present with Onivyde. And then there's other forms of irinotecan, like this one, which has a very hard to pronounce name, sacituzumab govitecan. And what this is the active ingredient of irinotecan on an antibody drug conjugate against TROP2, which is a common breast cancer marker. And this agent has a lot of activity, but unfortunately, even though this is highly expressed on breast cancer, this is how it traffics to the breast cancer cells. It's not expressed on sarcoma. And we know that most sarcomas actually don't have something on the surface that is common to, all Ewing sarcoma, for example, that we can traffic something in for. So this is a very good idea, but unfortunately, it doesn't really work for our population. So PEN-866 comes from another kind of class of kind of drug delivery agent called an HSP90 inhibitor drug conjugate. And it's kind of a mouthful, but I'll give you a little bit of a background on it. So HSP90 inhibitors were these drugs that were tested in clinical trials that were really not very effective as anti-cancer agents. This is back in the maybe mid 2000s, early 2010 time. So they didn't really shrink tumors. They didn't work really against any kind of particular cancer. But what was discovered about them was that they had what we call a pharmacokinetic advantage in cancer cells. And sort of what this means is that these drugs, for whatever reason, were held in tumor cells for longer and at much higher levels compared to normal cells. So as a sort of illustration of this, the drug would be trafficked into the normal cell and then trafficked right out. And this was usually in a matter of a couple of hours, maybe 5 hours. In contrast, in the cancer cell, it would be trafficked in, and then for the most part, it would be held in there for quite a long time, like on the order of days, 5 days, 6 days, 7 days, out to 10 days. And so once this was discovered, the company developing this realized, well, even though the HSP90 inhibitors were not effective, perhaps we can use this as a way to deliver more active drugs directly into the cell and keep them there. And so HSP90 inhibitor drug conjugates actually combine an HSP90 inhibitor so that you get this effect in the cancer cell stuck onto or conjugated to a payload or an active drug to deliver it into the cell. And so PEN-866 is one of these. So what it's comprised of is that in the blue, I'm showing you like an HSP90 inhibitor. In the yellow, this is SN38. Remember, this is the active metabolite of irinotecan. So it's inhibiting that repair of the two strands sticking back together when you have the supercoiling effect. And then this red thing, which is just a connecting kind of ligand. And the idea behind it was that it would act as an extended release kind of tumor-targeted chemotherapy with less effect on normal tissue. So it would achieve kind of all of the aims that we're looking for, less toxicity on the normal cells and more toxicity and more cell killing on the cancer cells. And the big advantage that it has over the ADC is that one I showed you before with the long, hard to pronounce name, is that it doesn't require anything on the surface of the cell to work. So you don't have to have necessarily a tumor that expresses a certain protein in order for it to work. We don't completely understand what it is about the cancer cells that hold the HSP90 inhibitor in there for a long period of time, but it seems to be universal, at least in the sarcoma models that we have tested. So that's a huge advantage. Okay, so the first sort of sets of experiments we did were to kind of try to understand how PEN-866 looked different from irinotecan and then also from just an HSP90 inhibitor. So this drug, gantenerumab, is just an HSP90 inhibitor, the ones that didn't really work in the clinic. And we kind of wanted to understand, well, do we see a difference when we look at this in mouse models? And so we started with a cell line xenograft model of Ewing. So here we take a human derived cell line of Ewing sarcoma and we inject it into the mouse right at the surface of the bone. And in this mouse experiment, we decided we were going to start with a really large tumor size because that's how our patients present, right? The patients don't come in with microscopic tumor, they come in with a big mass. And so what that looked like in the mouse, and I don't know how familiar anyone is with looking at mouse legs, but this should be like a little skinny leg. This is actually this very, very large tumor. And just as a test, we just gave one dose a week of each of the drugs twice. So we gave gantenerumab once a week times 2 weeks. We gave irinotecan once a week times 2 weeks. And then we tried 3 different doses of PEN-866. And I should say that the irinotecan dose is like the highest dose that you can get achieved in a mouse. And what we saw initially was not particularly surprising. So the blue is showing you a vehicle. This is just a mouse treated with essentially like saline, so it should grow. And this is the tumor volume over the number of days. And the little arrows here show you when we dosed. So here was our starting 1,000 millimeters cubed. We did our first dose, we did our second dose, and actually within like a week and a half, all of the vehicle mice had reached endpoint. They were too big and we couldn't continue the experiment with them. The gantenerumab, the HSP90 inhibitor, did exactly what we saw it do in clinical trials, which was nothing at all. So those tumors grew at the same rate as the vehicle. But really kind of interestingly, all of the other groups had tumor shrinkage. So the mice treated with the standard irinotecan, the tumors basically completely disappeared after the second dose. And the same was true for all 3 of the doses of PEN-866. And when I did this experiment, I was relatively new to the lab, so I didn't realize that most people just end the experiment there and call it a day. I just waited and I left the mice to see what would happen. And my boss at the time was like, after I showed him this data, was like, why did you do that? Nobody does that. But I honestly didn't know any better. And what we saw was really very interesting. So the purple here is the irinotecan. So we didn't dose the mice again after that second dose, and every single mouse had a relapse. So they all had tumors return and grow to endpoint within about another 30 days from the original start. But in the PEN-866 groups, we saw a further prolongation of survival in the mice that received the lowest dose. So they survived out to about 80 days. And then in the mice that received one of the 2 higher doses, we actually saw no recurrence out for 6 months. So this was an extremely long experiment. This is a long time to keep a mouse and look for the tumors to come back. And this is a really remarkable finding. This, we were really, really kind of surprised and intrigued by this. In the clinic, because we know that the longer you expose a tumor to irinotecan, the better it works, we actually use something called a protracted schedule. So if anyone has a child who's been treated with irinotecan, we don't just give one big dose once a week. We give 1 dose every day for 5 days, and that's a cycle. And so we thought, well, maybe if we challenged this PEN-866 drug against a protracted irinotecan schedule, it wouldn't look as good. So if we give the irinotecan the way we give it in the clinic, maybe it looks the same as PEN-866. So we did a very similar thing. We used a different model. This one was called EW8. Same deal, next to the bone, huge tumor size at the start. And we compared a protracted schedule where we divided that 1 big dose into 5 doses and gave it 5 days in a row for 2 weeks, compared to, again, just this once weekly dose of PEN-866. And we saw essentially the exact same thing. So the mice that received irinotecan maybe did a little bit better than the first experiment, but eventually by about 190 days, every single mouse had relapsed. The tumors had come back. And again, and here the drug had a different name, then it was called STA8666. But here we saw, again, none of the mice had a recurrence of the tumor. So again, a really exciting finding. And then we wanted to take it into a patient-derived xenograft model because we thought, well, sometimes cell lines don't behave like human tumors. And so this was a model of a tumor sample that was taken at some point from a patient and only ever grown in mouse models, so never was put on plastic and selected for different features. And we did essentially the same exact experiment. This was in a different background strain of mice, so we had to change the dosing a little bit. And then we also had a group here where we said, well, let's combine the irinotecan plus the gantenerumab to see maybe it's some additive effect with the HSP90 inhibitor. And what we saw, again, for kind of the third time, was that the mice treated with a PEN-866, now all of them did not have full, relapse-free survival, but about half of them had relapse-free survival out to about almost 200 days, whereas all the mice treated with irinotecan or with the combination or with the saline alone reached their endpoint around 120, 140 days. And then finally, we looked in additional sarcoma models, and I know this is Ewing's U, but I just thought it would be interesting to kind of show this data. We looked in a Rhabdomyosarcoma model. We essentially saw the same thing. You needed slightly higher doses and didn't see quite as much of a prolonged response, but for sure a difference from the standard agent. And then we did it in an osteosarcoma model, this was supposed to be our negative control. Irinotecan doesn't work against osteosarcoma. You can see that it grows right through. And even in this model, we saw the tumors shrink right down and sort of stay down. We wanted to understand whether what was happening at the level of the mouse tumors was what was happening sort of molecularly inside the tumor. And so we took Ewing's xenograft tumors again. We treated 1 time, and then we collected the tumors from the mice at different time points to look at whether we were getting prolonged activity of anti-topoisomerase 1. And so what I'm showing you here, these are this is a Western blot. So we're detecting different proteins here. And this protein is a protein that shows you that the DNA is being damaged. And so these are tumors collected from 3 different mice on 3 days after a particular treatment. So this mouse got vehicle, which is saline. This mouse got irinotecan, and this mouse got PEN-866. And you can see at the 3rd day, there's about a similar, maybe there's a little bit more DNA damage in the mouse that received the PEN-866, but the irinotecan mouse is still having some DNA damage in the tumor here. What was really more interesting to us is as we went out, and this is the day 7 data, and we actually have this out to day 10 as well, is that the tumors that were treated with irinotecan no longer are showing us that the DNA is actively being damaged. But the mice treated with the PEN-866 did have active damage of DNA all this 7 days out from their treatment. And then this is data not generated by our lab, but generated by another lab, where they actually looked at normal tissues and they compared vehicle to irinotecan to PEN-866. And this was really interesting data because if you look at sort of kidney, liver, or pancreas, you see a lot of DNA damage in the organs that received irinotecan. So 30% of the cells had these positive nuclei, which shows active ongoing DNA damage, compared to the PEN-866 animals, which had less than 5%, which was very similar to what was in the saline-treated mice. So this really suggests, and this was 7 days after dosing. So this suggested that in normal tissues, you were not damaging the DNA, but in the tumor tissues, you were. And then this was just an experiment that we did just to look at kind of what genes are changing, what expression of genes are changing with irinotecan treated tumors versus PEN-866 treated tumors. And what you can see is actually the top 10 genes that went up expression and the top 10 expression of genes that went down were exactly the same between the PEN-866 and irinotecan treated animals, but the magnitude of those changes was much larger, in particular in the downregulated genes for PEN-866. So again, this I think is evidence that this drug is working by the same mechanism of irinotecan, but just in a much more potent way. Okay, so that was a lot of preclinical data, I know, but just to sort of summarize, we know that in animal models of sarcoma, treatment with PEN-866 resulted in prolonged exposure and increased DNA damage in tumors compared to the standard drug that we use, irinotecan. In mice, we saw less damage to normal cells compared to irinotecan using the experimental agents. And we saw that the drug was active in both xenograft and PDX models of multiple different pediatric sarcomas, in particularly in Ewing sarcoma, where it had the most activity and was superior to that of the standard agents. So this data was all very exciting. Actually, the company developing the drug was very excited by this drug. And so it moved pretty rapidly into a first-in-human adult phase 1 clinical trial. And I'm going to share a little bit of that data here. So it started in August 2017. So again, we're still now like, how many years ago? 8 years ago. And they did this phase 1/2 kind of multi-center study to look at safety and some of the preliminary anti-tumor activity of this drug. It was first in human, which means they were only allowed to enroll patients 18 and older, and it was sponsored by the drug company. And they looked at PEN-866 all by itself, so just single kind of monotherapy in a bunch of different escalating dose cohorts, and they treated 3 times every 4 weeks, so once a week for 3 out of 4 weeks. And I'll kind of skip to the results. They treated 30 patients in 8 cohorts on the phase 1. Of course, there were dose-limiting toxicities, meaning toxicities experienced in patients, and that was in some of the later cohorts. So they had 1 patient with pretty severe diarrhea, and then some patients with dehydration and fatigue as they got to the highest doses. And then they established this dose, 175 mg per meter squared, as the maximum tolerated dose for single agent use in adult patients. And so any drug is going to have adverse events. The common ones were low blood counts, some GI side effects, headache and fatigue. So it definitely looked similar to irinotecan, but I will say that the severity of these adverse events were substantially less than what we typically see with irinotecan in adult patients. And then of these 26 patients that were treated at all different doses, they had one patient with a confirmed partial response. So that means that the tumor shrunk down more than 30%. They had 6 patients with what they call decreased target lesion size. So that means it shrunk, but either they didn't get a second reading to determine how much it shrunk, or it was less than 30%. And then they had 11 patients with stable disease, meaning it didn't grow more than 20% over the period of time. So of this sort of group of 26, you had about 18 that had some kind of response to the drug, which is pretty good for a phase 1 study. More interestingly is they had 2 patients who agreed to get biopsies on this study. And so they had one patient was biopsied before and then on day 2, and another patient was biopsied before and on day 7. And they looked at levels of PEN-866 in the tumor and in the plasma at 24 hours and then also at 7 days. And this is really remarkable. I know that the numbers may not mean anything to anyone. What does 318 nanomolar mean? But basically the punchline here is that in the tumor, you had a very high level of PEN-866 and a very high level of SN38, even at 24 hours. And just to give you a little bit of reference, when we treat Ewing sarcoma cells in a dish, we need less than 1 nanomolar to kill them of SN38. So 86 nanomolar is quite a lot at 24 hours. And then the other kind of very interesting finding was that in the plasma, there was actually less than what was in the tumor, which is what you would expect to see. So what was circulating around the body and hitting all the normal cells was substantially less, like 40-fold less than what was hanging out in the tumor. At 7 days, you saw this even more. So this is, again, after just a single dose. You still had 2 nanomolar of SN38 in the tumor, which again is more than you need to kill cells in a dish. And the amount of this in the plasma was below the level of detection. So it was not really circulating anymore. And again, these were really consistent with our preclinical data where we saw this retained drug releasing into the cancer cells over multiple days. Okay, so then it was time for our study. So the rationale for our study is kind of, as I said, so prolonged exposure to TOP1 inhibition is better in pediatric type sarcomas. So more doses over more days is better than 1 big dose. And again, this is why I told you our current regimens give irinotecan every day for 5 days for 2 out of every 3 weeks to kind of get the best bang for our buck. We know that from our preclinical data, the PEN-866 is acting as a delivery agent, and so it facilitates this prolonged topoisomerase 1 activity. And we know that in our models, at least, the activity of PEN-866 was better than irinotecan. We also know that very rarely do we treat sarcomas with just 1 drug at a time, and we know that irinotecan is commonly combined with vincristine and temozolomide as a highly active regimen in relapse for patients with Ewing sarcoma that's come back, or in rhabdomyosarcoma. And so the idea behind this study was to determine essentially the safe dose of PEN-866 that can be combined with these 2 other chemotherapies in patients 12 and up, up to 39 years inclusive, with relapsed or refractory solid tumors. And we chose these age ranges because we were permitted by the FDA to use the adult data if we started at 12 and went up. And then 39 years is the definition for a young adult, and that's why we stopped at 39. And then we have a bunch of secondary objectives, kind of looking at similar things that the phase 1 study looked at, how much of this drug is in the plasma and in the tumor, and really to describe the toxicities of this triple regimen, which have not been described before. And then we have a phase 2 part of this study, which is to ask the question of, is this actually working? So to look at the response rate, so patients who have complete tumors go away or partially go away in the combination over a period of a year or 18 treatment cycles. And then, of course, we're looking at the length of survival without disease progression and some other analyses to look at the degree of DNA damage in matched pre and on-treatment tumor and normal tissues for patients who consent to biopsy. This next slide is really overwhelming. These are our sort of lab correlatives. So these are the kinds of other questions that we're asking of patients on this trial. We're looking at other markers of how the drug is working in both immune subsets, tumor, and normal cells. Looking at the relationships between the amount of drug that's getting to different compartments and how well the drug is working. Looking at circulating tumor DNA and changes in that over time. Looking at immune markers and some particular expression of certain genes that we think may have relevance to these particular drugs. And then finally, some quality of life and cognitive functioning and emotional well-being aims, like PRO kind of related aims. Okay, so I'm almost done with my slides. So the study design is a dose finding for phase 1. So we're enrolling groups of 3 patients at escalating doses. And for we're in the phase 1 right now. So in terms of eligibility, we can take actually any patient ages 12 to 39 with any relapsed or refractory solid tumor, including patients who have received like a VIT or an IT combination before. And then in the phase 2, which we hope to be enrolling, as soon as we finish the phase 1, we have specific expansion cohorts for Ewing sarcoma and rhabdomyosarcoma. So these patients can enroll in the phase 1, but other patients can enroll in the phase 1 as well. But in the phase 2, it's specifically for Ewing's and rhabdo. And we're using what's called a 2-stage design for each cohort, which means we enroll a small number of patients, get a preliminary sense of whether the combination is effective. And if it looks like there's enough response, then we enroll a larger number of patients for each of those cohorts kind of separately. And the eligibility here is similar. It's just instead of all solid tumors, it's Ewing's or rhabdo. And right now we have an exclusion for prior therapy with temozolomide combinations. And this is what the schema looks like. So patients come here. This study is open right now only at the NIH. Patients come here on day 1 and day 8 for the vincristine and PEN-866 infusions. And then the temozolomide they can take at home for 5 days. That's oral. On day 15, they can see their local provider and get labs and an exam, which gets sent to us. And then they come back to us on day 21 or day 1 of the next cycle. And the doses of vincristine and temozolomide stay the same. And we increase the dose of PEN-866 like from a patient to patient. And these are the doses again. So our starting dose is this 125, which is just 1 dose level below what was the adult recommended dose. So we think we're at least close to the range or within the range of where we hope to see activity. And then I don't need to really show you the statistical plan, but this is what I was talking about before, this 2-stage design. So we would start with a small number of 9 patients in each cohort to sort of minimize the exposure to patients who may not be benefiting. So if we don't see, enough response in the first stage, then we don't enroll additional patients. And that's because it's a rare disease and there aren't a ton of patients. So this was our original study design, and now it's 3 years ago in March 2022, we had completed all the regulatory approvals, which was 5 years after the adult study, which still feels like a long time, but actually the timeline was pretty good. And then a week later, the industry sponsor filed for bankruptcy. And so we had no way to access the drug. And so this was kind of like how it felt. But I will say since 2022, we had a lot of help from the community, and this is sort of my favorite part of the story. So we worked with Nancy Goodman at Kids v. Cancer, who's an incredible advocate for pediatric cancer drug access and was responsible for the RACE Act getting through and is just a very persuasive patient advocate. We worked with folks from the FDA who kind of knew ins and outs and ways that we could maybe revive this trial. Folks here at NCI, and we basically petitioned the lien holders of the previous industry sponsor. So these were the people who had sort of the financial rights to the drug and financial rights to the company, even though the company had been dissolved. And we said, look, we know that you have this investigational agent in a clinical drug supply, and we know no one's going to use it. Will you give the drug supply to the NCI to run this study? And after much back and forth, they agreed to do so. So right now, NCI has the rights to the actual existing drug and the actual existing drug. We don't have the rights to the intellectual property, but we have the drug and and have permission to use it in the study. And so we opened to enrollment in November, and we're now actively recruiting into the phase 1 part of the study. And so I think, I really for a long time felt like this is a model that maybe we could use again and again as we see drugs kind of be orphaned or abandoned by drug companies. If we can do this one time, maybe we can use this as a model to continue doing it. Because again, the goal, I think, on the clinical side is really to increase our access to test new agents, which will allow us to increase the potential for improvement in outcomes for patients. It doesn't matter how much we know about the lab science. If we can't get the drugs to test in patients, we can't make progress. And so I will stop there to leave some time for questions, but I just want to thank the members of my lab who did this work, a ton of collaborators, both within the NIH and outside, the consultants who helped work to get this drug, moved over into a place where we could actually use it, even though the company was no longer solvent. And then, of course, the patients and their families who donated their time and energy to the initial phase 1 adult study and our current study, and then my funding sources. And so I'm happy to take any questions.
Great. Thank you so much. You did a really beautiful job explaining that in a way that was easy to digest and understand. Really exciting and informative. Can you tell us a little bit more about, there haven't been many Ewing sarcoma trials through NIH. What does it look like for a patient to go to the NIH for a trial?
Yeah, no, that's a great question. I will say it's a little in flux right now, what it looks like. But for the most part, so the NIH is entirely taxpayer funded, and it's the largest clinical research hospital in the world. So basically, every patient who comes to the NIH for treatment is a patient who's enrolling on a clinical trial. And that means that we don't sort of engage with insurance companies. The study drugs, the care, admissions, office visits, supportive medications, all sorts of those things are covered. Patients don't have to pay anything out of pocket. Basically, the entire care is covered. And then, for our disease population, we don't, everybody is not within 20 minutes driving. So many times we're having patients come from farther afield, and we cover travel and accommodation cost for patients once they've started therapy. So we have an inn on campus where patients and families can stay, where they have like a community and provide meals and shuttle buses to grocery stores for kind of long-term patients and stuff like that. And it's literally like right down the road. And so patients can walk up if they need care or anything. So we try to make it as patient-friendly as possible because it's, obviously we're meeting patients at a difficult time in their therapy. Most of the patients who come here are those who have exhausted standard therapies kind of across the board in cancer and in other diseases. But that's essentially how it works.
Great, thanks.
Awesome. Thanks for that. I want to touch a little bit on the preclinical science, which you did a great job of breaking down. And I couldn't help but notice you tested in Ewing sarcoma, rhabdomyosarcoma, but it seemed like a pretty positive indication for osteosarcoma. So are there any plans to expand that to that group as well?
Yeah, so that's a great question. So we didn't feel like we had enough data with our one model in osteosarcoma and one mouse to say we're going to open a cohort in the phase 2. That said, we would love to recruit osteosarcoma patients in the phase 1 because we did have this little carve out, which is such a detail that I didn't put it in the slides, that if we had patients with other types of tumors that showed an interesting response in the phase 1, that we would open other cohorts for them. So and again, like the statistics are not perfect, maybe we wouldn't have quite enough to show an effect, even if there were an effect. But like if we enrolled an osteosarcoma patient in the phase 1 and we saw a nice response, I think that would be enough rationale to say, okay, we're going to open a little cohort for the osteosarcoma patients and add them into that phase 2. We're a little, the tricky part about this not having an industry sponsor is that we have finite amount of drug. And so,
Right.
I would love to say, like, let's open it to, more patients. And, if we see partial responses in some, or even if there's a little preclinical data in some, anyone who wants to come on this study should be able to test it. But we sort of run the risk of running out of the drug supply. And so that's kind of how we're approaching it now. But if anyone has an osteosarcoma patient or knows an osteosarcoma patient who's looking for something, we are happy to enroll those patients in the phase 1. They're allowed on the phase 1.
Awesome. And the reason I mentioned that too, I noticed several attendees are from MIB Agents, which is an osteosarcoma patient advocacy group. So maybe you can take that conversation offline and discuss what that could look like in the future.
Absolutely. Yeah.
Great. When I was preparing for this, I saw that there are some more common adult cancers where this drug is being tested as well. And how does that work with the kind of finite supply of it when, something much more common like colorectal cancer is investigating this as well? How do we protect the pediatric population and make sure that they get a seat at the table?
Yeah, so we almost never get a seat at the table. And I left this out of the story, but I'll tell you this part of the story now. So when, we had done all this preclinical work, and the lead site for that first in human study was here at NCI, and it was run by a lung cancer doctor. And so we, for sure, were trying to elbow our way in and have a seat at the table. We were at all the planning meetings, et cetera, et cetera. We lobbied at the time of the phase 1 for a sarcoma cohort in their phase 1B. And we said, it's fine, even if you don't want to drop the age, like, please just give us a sarcoma. We'll make it an all-comers sarcoma cohort. Like, just please give us the opportunity to enroll these patients. And they said yes. And so we were planning for that. And then before they got to that phase 1B, they came back to us and they said, well, we have the phase 1A data now, and we have a couple of, types of cancer that we already saw good responses in. So we're going to shift our priorities, and sorry, we're not doing the sarcoma cohort. And I can't tell you how often that happens, where we have like initial buy-in at the beginning, and then they have limited resources too. They are all for-profit companies, and so they basically make the calculation that makes the most sense for their stakeholders. Where we are now, because the company is bankrupt, we are not competing with anybody. So there are no adult patients actually getting this drug. There is no open adult study for this with this agent. We are the only ones who have it, and so it's like a plus minus. I think it would be better if there was an industry sponsor to help kind of shepherd this along, but we kind of own it all for now. So we're not competing at the moment for this limited drug supply. It just belongs to us.
And is the thinking there that, God willing, this trial is successful, that pharma will re-engage
Yes.
manufacturing?
That is the thinking. I mean, that is the hope. And people ask me that, like, well, what are you going to do if this works? Because you're still kind of like up a creek and you don't have a sponsor. And, my approach is we can't let the like negative what if stop us in our tracks before we get there, right? We have to just take it one step at a time. There are a lot of incentives for pharmaceutical industry companies to take drugs that have no other indication and make the indication first in pediatrics. There's a huge financial carrot that is provided if that is the case. And it was supposed to sunset in the fall, and I believe it got extended. And so, I think if we were able to show like, look, this works in this disease, now someone needs to run a registration study for an indication, I think that there would be pharmaceutical interest because there's, money at the end of it. Yeah.
Excellent. So you kind of alluded to this since supply is finite, but there are a couple questions here asking if other locations will ever be opened.
Yeah, so this is originally when we had the industry sponsor, it was a multi-center study. That was the plan, to have it to be a multi-center study. I think now, both with the fact that we're running it without an industry sponsor, and then also sort of the climate in terms of like governmental resources, it's actually really resource intensive for the government to open a study at multiple sites. And so we've essentially been told that we're not doing that right now. So unfortunately, we are just us. And, I know it's really hard. We get referrals from patients in California and Oregon and Texas. And I think, without being able to say like, this is a slam dunk and your kid is going to benefit from it, you should come, which of course we can't say because it's experimental. It's a huge ask to have people come, once a week for 2 out of every 3 weeks for however long. So I completely, sympathize with the fact that we would love to have this open in more geographic locations. And hopefully if things change, we can try, but it's not in the cards at the moment, unfortunately.
Right. Understood. Thank you.
Yeah.
Great. There is a question here that says, with the prolonged drug in tumor scene, is there a thought about watching how long it stays in the tumor and spacing out treatment further to help mitigate side effects?
Yeah, so that is an excellent question. So in our study for patients who are 18 and older, we are asking them to consent to a biopsy if it is safe and easy to do. And we are going to be looking at what is the drug doing in the tumor at different time points after the dosing, specifically for that reason, so that we could either space it out or maybe we need to make it closer, depending on how things look. I will say, we've only enrolled a very small number of patients, and the statistics, it's just impossible to say like this is what we see. But I will say that in the small number that we've enrolled, the side effects do not look at all like what we typically see with kind of standard chemo regimens. We've seen some lab decreases in blood counts, but no one has needed transfusions. We haven't had anybody need to be readmitted. This is a totally fully outpatient regimen. So I think, hopefully this dosing schedule actually already mitigates the side effects as it is. But I do think that, having that biological data from the tumor is really invaluable. And as you can see from the adult phase 1, I mean, they only had 2 patients consent to biopsy and they got a ton of information from that. So one patient can give us so much information, which is why we're asking the 18 and older patients to consider that on this study.
Thanks for that. I kind of want to build off that and go back to like the mechanism of action, if I understand this correctly. So there's better pharmacokinetics in the tumor cell, meaning longer residence time of this drug conjugate. Why do we think that is? Is there more of the HSP? Are those heat shock protein 90s?
Yeah.
Is that what it is?
That's a great question. This is like the question that you get at all the scientific meetings. So we think that it has something to do with the isoform of the HSP90. So we think there's different kinds of HSP90 that exist in different types of cells, and the amount of stress that a cell is under can change the proportion of those different isoforms. So we think that we know, I mean, from like decades of other people's research, that cancer cells are under an immense amount of stress. They're kind of stressed all the time because they're like living in environments that are hostile to them, low oxygen and et cetera. But we think that that's most likely contributing, that they just have a different sort of mix of the HSP90 isoforms than what the normal cell has, and that particular mix makes them hold on to it longer. What we don't know actually is whether that's true for every single kind of cancer. So there may be types of cancer where there is no pharmacokinetic advantage. And I will say that most of the cancers that have been looked at preclinically are typically like pretty aggressive, fast growing cancers. So like lung, colon, sarcoma, cervical, whether that's maybe different in a low grade type of tumor could be the case. I don't think anybody's looked at that.
Okay. Thanks.
Mm-hmm.
Is there consideration for an upfront therapy or a maintenance therapy with this agent?
Yeah, so I think we have to get these data back first and see what we see. But I think, we know that irinotecan is actually already used in the upfront setting for some types of rhabdomyosarcoma. I think if you saw activity of this, agent that exceeded or was equal to the standard, I think moving it up front would make a lot of sense. And I know that, in the Ewing's community right now, we don't use irinotecan up front. We just typically do a VDCIE, but there are groups that are kind of advocating, and there's some data that I think is percolating around that maybe irinotecan in the upfront setting would have benefit. And so I think that would be a great study to run at some point. We'd have to really prove that it's better than the standard, but for sure, I think moving something that looks active up front is the way to go. There's just a lot of bars to cross before you get there.
Absolutely. I've got a logistical question here for somebody that's in Europe. is this trial open to European citizens, assuming they're able to travel?
So I'm happy if you want to contact me offline. It's very complicated with international travel because the way that the government handles the travel costs is extremely different from domestic travelers. But feel free to email you. If you Google me, you can get my email and email me, and we can kind of talk more specifics. I'm happy to answer that. It's complicated.
Okay.
And if you can speak to it generally, there's a question here about concerns with cuts to your department, and do you have a high degree of confidence of this trial um, of this trial
Completing?
Yeah.
Yeah. I mean, I hope so. We are all still here. So I'm in the pediatric oncology branch. All the pediatric oncologists are still here. Like the clinical infrastructure in the clinical center is still here. I mean, for sure, there have been some challenges over the last couple of months. And I feel like every day is kind of a new work in progress, trying to figure out workarounds and things like that. But I do think that, there is, I think pediatric oncology in general, and in particular, like pediatric oncology treatment, has general public support. And so I'm really hopeful that, that general public support, which we have seen kind of come to bat for us in many other instances over, the last many years, will be able to kind of sustain what we're doing. So I'm really hopeful. I don't know, but I'm staying here until they fire me. So.
Awesome. This is a little bit of a technical question. I don't know if there's any way to answer it, but I'm going to ask it anyways. Do you have any idea how this drug conjugate compares to the recently opened PL224 trial, which is a different topoisomerase inhibitor?
Yeah, no, that is a great question. So, and I'm glad, the PL224 story was kind of like, we've been like back and forth with them. And I know a bunch of those investigators as well. I don't think we know. We haven't tested it in animal models. And even if we had sort of done a head-to-head in animal models, I think it's so difficult to know like how that translates into patients, because we don't really know how the tolerability of each drug in a mouse, for example, is going to translate into a patient. So something that is very highly tolerable in a mouse may be less tolerable in a patient, and you may be able to only get, to a lower dose, and vice versa. You might have something that you can't use very high doses in a mouse, and in the patient cohorts, we can go up a lot higher. So I really think we just have to kind of look at the results of the two studies when they're done, and then make a decision. I mean, and if they both look, I mean, the sort of dream would be that they both look good, right? Because the biggest problem is that we don't have anything to take forward. So I'm rooting for PL224 and PEN-866, and we'll just see. But I don't know the answer to that question.
Right. Thank you.
Yeah.
Great. There are a few specific questions that came in about eligibility. I would encourage those folks to check out the clinicaltrials.gov and reach out to Dr. Heske's team if you have any specific questions about your eligibility. Pierre, do you want to take this last question, or should we wrap it up from here?
I think, this is kind of open-ended, and you kind of touched on this already. It's a great way to end, I think. In the next 20 years, where do you see Ewing's sarcoma treatment going? Do you see change coming to treatment at initial diagnosis? And what seems promising to you? I mean, I think this story is very promising to all of us, but beyond that, do you have anything for us?
No, that's a great question. I mean, if we don't see any change to upfront therapy in 20 years, we are really, really not doing a good job. I think, we really haven't seen a huge change in the last 20 years to upfront therapy. And I would hope that, we would see something different. The reality is, Ewing's is a very chemosensitive tumor in a lot of cases. We have a lot of patients who are cured with standard therapy, but the problem is all of these side effects, short and long term. And so I think, we probably have to do some combination of not completely reinventing the wheel, but like tweaking what we have so that we're either combining the therapies in ways that we're able to use lower doses that don't cause as much toxicity, or deliver the drugs in a better way, so that we're not sort of hitting the normal cells as much, plus potentially some novel treatments coming through. I mean, there's a lot of effort trying to target the driver. You know, EWS-1, there's obviously, everybody knows about the trabectedin story where you have, it's a chemo, but it's also sort of targeting EWS-1. I mean, I think that people are doing a lot of really interesting work in this space. But again, I feel like the real bottleneck for us is that even though there's a lot of stuff going on in the labs that's really interesting, it's extremely hard to bring that forward. And so like my kind of side hustle pie in the sky idea is that we can change something in the operational way that we integrate ourselves with the pharmaceutical industry and how we are able to, get these sort of lost drugs back off the shelf for testing.
Yeah, absolutely. The business side is often the story. I feel like gets left out, and something that we were totally naive to as a foundation, but so important to be able to span the gap between what happens in the bench to the bedside. But we want to just thank you for your time and your perseverance and your grit to bringing this trial to us and making it a reality. It's like truly captivating. So thank you so much for all that you do, and thank you for being here with us. I am going to bring the session to close. Thank you to my fabulous co-host, Emmy, for joining me as always. It's a pleasure to have you here. And lastly, thank you to our viewers for joining us today as well. Please remember to stay up to date, subscribe to our Ewing's U emails. The easiest way to do that, go to littlewarrior.org, navigate to the Ewing's U tab on our website. We do not have anything scheduled, so if you have a guest presenter or speaker that you'd like to see, we'd love to hear about it. Shoot us an email at info@littlewarrior.org. We'd love to hear more. And also follow us on Instagram and Facebook, as we always post about our next speaker that way as well. So that's it for now. Hopefully you enjoyed this episode. We'll see you here next time. Thanks, everybody. Swords up.
Thank you.
Swords up.
Swords up.